Let me introduce about MSI team, one of the research teams in the Fukusaki laboratory. MSI stands for mass spectrometry imaging. This video introduces the outline of mass spectrometry imaging and research results in the Fukusaki laboratory. Seeing is believing. This is the key phrase for the MSI team. From long ago, researchers would like to see what we cannot see using new technologies. As the saying goes, seeing is believing. Visual information is intuitively understandable compared to complicated numerical information. One of conventional technology is microscope. Using microscope, you can see structures, but you cannot find components. Imaging mass spectrometry is one of molecular imaging technique using mass spectrometry. Using this technique, you can see many kinds of components without labeling. This is an overview of mass spectrometry imaging. Mass spectrometry imaging is a two-dimensional method of performing mass spectrometry on tissue sections. First, body tissues such as mouse brain and plants are sectioned. Molecules in the tissue are ionized by irradiating the tissue section with a laser for mass spectrometry. By repeating this on a two-dimensional tissue section, the position information and mass spectrum on each position can be obtained. Then you can select the peak that appears at M over G of the target compounds and obtain the distribution image of that compound. Mass spectrometry imaging allows us to know what kind of substance, where, and how much exists from this image. This is an example of using mass spectrometry imaging on hot pepper. As you know, when you eat hot pepper, you can feel the spiciness of capsaicin, which is a pungent compound. However, it is impossible to know which part is pungent just by looking at the organizational structure of the capsicum as shown on the left of the photo. Therefore, we use mass spectrometry imaging to visualize the distribution of capsaicin. Then, two peaks derived from capsaicin were observed in the mass spectrum. By selecting these peaks and generating images, we found that capsaicin was accumulating in the placenta rather than in the pericarp. At Fukusaki Laboratory, IM-Scope QT, manufactured by Shimazu Corporation, is used for mass spectrometry imaging. IM-Scope QT is equipped with an optical microscope for observing the sample morphology and a laser used for ionization. Also, it can fuse with a mass spectrometer that measures the mass of ionized molecules. Then it is possible to superimpose and compare optical images and imaging results. On the development of applications and methods for a wide variety of samples other than those listed here. From now, I would like to introduce recent research achievements. The first is the development of a method for visualizing enzyme reaction on plant tissues in MSI. The localization of enzyme reactions in living organisms is important for understanding biological phenomena. Enzyme histochemistry has been used to study the localization of enzymes. However, there are many limitations, such as the need for staining and there are a limited number of enzymes that can be examined. By introducing mass spectrometry on the tissue, we could compensate for these shortcomings and have succeeded in development a new enzyme histochemistry method. In this study, we found high GAD activity in the vascular system in the hypocotyl and root tip meristem of germinating soybean seeds, which are also known as model plants. The second is enzyme activity imaging of insects. Many pesticides are used in the world, and they can have effects on more than just pests. However, whether neonicotinoids affect the distribution of enzyme activity in non-target organisms has not been investigated. Therefore, we visualize the effect of neonicotinoid on the distribution of enzyme activity by mass spectrometry imaging using the model organism Drosophila melanogaster. This analysis revealed that neonicotinoids inhibit enzyme activity in the fly head. The third is the metabolite imaging of rice koji. Koji is an important ingredient in the producing of sake, and it is said that the quality of koji greatly affects the quality of sake. The quality of koji is usually judged by observing the structure. However, little is understood about the metabolites that affect the surface structure. Therefore, it is important to reveal the relationship between the quality of koji and its metabolites. It is also necessary to prepare sections for visualization of koji, but it is difficult to make a cross-section because koji is small and fragile. In addition, the analysis of koji using mass spectrometry imaging is an unprecedented attempt. Therefore, the purpose of this study is to develop the mass spectrometry imaging method for metabolites imaging in rice koji. By using an cryofilm, it has become possible to prepare sections while maintaining the morphology of the koji. Furthermore, we succeeded in maintaining ion intensity and localization by supplying a matrix by sublimation. Choline and isohosphatidylcholine were discovered by MSMS analysis using this method and comparison with an online spectrum database. This year marks the 10th year of the MSI team. Many papers were published in 2024. If you are interested, please feel free to contact us.